My name is Michelle Worrell-Carrillo, and I'm going to talk today about a pharmacogenomics knowledge resource called PharmGKB. So PharmGKB stands for the Pharmacogenomics Knowledge Base. And the mission of this NIH-funded knowledge resource is to collect, encode, and disseminate knowledge about the impact of human genetic variations on drug response, or that is, pharmacogenomics. The group at PharmGKB, the scientists there, curate genotype and phenotype data from peer-reviewed literature, drug labels, and drug dosing guidelines. They also use the information from these curated resources to create drug-centered pathways and very important pharmacogene reviews. The group at PharmGKB also works closely with CPIC, which I will talk about a little bit later, to create genotype-based drug dosing guidelines. So this pyramid that you see right now represents the information contained in PharmGKB. They call it the knowledge pyramid. And at the bottom layer, the base, you can see it says primary pharmacogenomic literature. And that's because that's basically what everything in PharmGKB is based off of. They take the knowledge from that literature, they annotate it, aggregate it, integrate it together to create the annotations that you'll find on the website itself. They do clinical interpretations, and then the goal is to approach clinical implementation. PharmGKB itself is not involved in clinical implementation, but it does provide resources for clinical implementation of pharmacogenomics. So I'm going to describe the website to you today. I'm going to give you a demo of it. And I've got different sections. I've broken it out into different sections. So the very first section I'm going to talk about today is the homepage overview of the website and how to register on the website. Okay, so this is the homepage of PharmGKB right now. So there are basically three main ways to access information in this knowledge resource. You can search using the prominent search box on the webpage. You can browse the information, and you can download it. And I'm going to go through each of those different ways. Right here, the search bar in the middle is the fastest way to search for people who know what it is they're looking for in PharmGKB. So I'm going to come back to that. Downloads, this Download tab, is how you will get to download all the information. I'm going to go over that part at the end. But first I'm going to introduce you to the homepage itself and tell you a little bit about how to browse the knowledge base. At the very top, we have a few buttons that are kind of quick resources to get to. Publications, this is just a list of PharmGKB publications, which you can search by type. You can filter it by searching for something in particular, or if, for example, you're just interested in dosing guidelines published through PharmGKB or in conjunction with PharmGKB, you can click that box, and it will filter the list for you. Anytime you see this icon in the upper left-hand corner, you can click it and you can just go back to the home page. So no matter where you are on the website, you click that button and you're back to this page right here. The news button at the top will take you to the PharmGKB blog, and it opens up in a separate tab. And this blog is maintained by the scientific curators at PharmGKB and the leadership at PharmGKB. And it's got new articles out about several times a month. I'm going to go back to the home page. The contact, anytime you want to send feedback to PharmGKB, if you just press that contact at the top, you put your name in there, your email if you want a response. If you don't provide an email, the scientists at PharmGKB can't get back to you. And then your message, and then there's like a little question to make sure that you're not a robot, so that PharmGKB doesn't get spammed. Okay. In the main section here, if you scroll down, we have a few different ways to browse the information. I'm going to skip over that for just a second. This main part in the center of the screen is really an introduction for people who don't have a lot of knowledge about the field or the resource itself. So for people who are new, they can click these, either what is pharmacogenomics or tell me more about PharmGKB. And it takes you into a whole separate display about information about that particular topic. I'm not going to go into that in this particular demo. You can look through that yourself at your leisure, but it's new to the PharmGKB site and is really designed for, like I said, people who are really unfamiliar with the resource. So we try to address a lot of people at the same time. So the search box is for people who know what they want to do. Browsing is for people who have an idea of what they want to do. And this part is for people who have no idea what they want to do yet, and they just want to get some more information about the resource. If you scroll down the home page, you'll see other resources. You'll see a recap of the latest news, which is just the most recent issues from blog post. Links to some partners of PharmGKB. These will take you off the site to CPIC, PGRN, or Precision FDA. And at the bottom of the page, we have more information about PharmGKB itself. If you want to learn about the team or the policies, how to cite PharmGKB, all those links are at the bottom. We do have an FAQ link as well. This is a great place to go if you have questions about particular source of information on PharmGKB or type of data that is found on the website. Most of the information on PharmGKB is just completely available up front. Everything is free at PharmGKB, but there are certain types of information where you do have to sign in. You have to register and sign in order to be able to see them. So I'm going to take you through the registration process right now. It's really simple to register. It doesn't take a lot of time. You don't get spam. But like I said, in order to see some of the information that I'm going to show you in this demo, you do have to be signed in. So if you don't have a user account already, you go to register. And you have to read through the policies. Basically, you're agreeing that you're not going to redistribute the information on PharmGKB or use it in any nefarious way. Step through the agreement process, and then you just enter your email. I'll just give you an example. Some kind of password, any kind you can think of. You go through basically the whole thing. I'm not going to fill everything out for you right now, but you go down to the bottom and you hit sign up. When you do that, you will get an email sent to you automatically. There's a link in the registration email that you have to click in order to get your account activated. So please, if you do this, check your junk or your spam folders if you don't see an email within several minutes of registering. But once you find that email, if you don't find it, you can go to the contact button on PharmGKB and send us an email so you didn't get it. We can resend it. But you need to click that link in order to activate your registration. So now I'm going to sign in so that you will be able to see all the information as I demo it. Okay, so now I'm signed in, and I just wanted to point out that your account information is right here. If you ever need to edit any information about yourself or change your password, you can do that easily by clicking these links under the account button at the top. Okay, so in this section, I'm going to talk about the types of information at PharmGKB and how to browse that information through the website. I'm going to take you back to the pyramid that I showed in the previous section. This pyramid is now broken out, and you can see at each layer of the pyramid, there are different types of information if you follow those arrows. Basically, we annotate the literature, including primary experimental literature, guidelines about how to dose drugs based on patient genotypes, and drug labels, like FDA drug labels or drug labels from other agencies. The scientists at PharmGKB create summaries of that pharmacogenomic information based on the annotations, and including pathways, VIPs, and they also create summaries about the pharmacogenomic information. And another process I mentioned earlier was creating these dosing guidelines. Since PharmGKB is a pharmacogenomics knowledge resource, it sees the world in a few basic chunks. Genes, the variants within those genes, and haplotypes that are in those genes. Haplotypes are combinations of the variants in the genes. Drugs, phenotypes, and the literature that things are based on. So those are like the basic entities in PharmGKB. Each one of these types of information has their own pages in PharmGKB, which I will be showing you. The other entities in PharmGKB are the annotations that the scientists create based off of the literature that they curate. So there are literature annotations, variant annotations, the pathways, etc. So I'm going to be discussing each of these types of entities in PharmGKB as well. So these boxes at the top are quick ways to browse the things that are probably browsed most on PharmGKB. So out of those entities that I listed, the drugs, pathways, dosing guidelines, drug labels are some of the most sought after pieces of information on PharmGKB itself. For drug, and there are little counts here. There are icons and little counts next to each one of these data types. So there are more than 618 drugs represented in PharmGKB, but 618 of them have some type of annotation on them. So if I click this, basically you are brought to a list that you can browse alphabetically. So there are a lot of, too many drugs to look through, 618, so we break it down. So if you're interested in aspirin, you can search under the A, or you can just type in the information as well. When you're browsing the list of drugs, you can see underneath each drug name the types of informations, dosing guidelines, drug labels, etc., that PharmGKB has for that particular drug. So in that way, you're browsing through the list of drugs, seeing what kind of information has been annotated on them without having to drill down to a specific drug in and of itself. Pathways on PharmGKB are quite popular. So I'm going to go to the pathway landing page. There are a little over 100 pathways on PharmGKB right now. They're all listed on this particular landing page. Most of these pages, these browsing pages that I'm going to show to you today, have some kind of chunk of information at the top that you can read about them, about how they're created, and what they're comprised of. I'll leave you to read that on your own. But I'm going to show you some of the functionality of these types of pages. So on the landing page here for pathways, you can, if you want to group pathways by certain types, or you want to isolate certain types, say gastrointestinal agents, you can just click that box. It'll filter the list for you and just limit the list to those couple of pathways. You can combine, if you want, are you interested in those and another type of pathway. You can click and choose however many you want in that way. Also, if you have some idea of a drug name that you are interested in for your pathway, you can type it in the filter and it narrows the list for you. So I'm going to show you one particular pathway, warfarin pharmacodynamics. So I'm just going to click on that pathway right here. Most of the pages that I'm going to show you have a side nav on the left-hand side, and this is an example of one. It also has a couple counts at the top here about different kinds of information regarding this particular pathway. So at PharmGKB, pathways are created by the scientific curation staff based on the primary literature. So they do pretty much full lit reviews of a particular drug, looking at either metabolism or mechanism of action or both. And then they come up with a display, some kind of picture representing the drug and the genes. Remember I said in PharmGKB we focus on genes, but it's really the protein product of the genes doing the action most of the time. But we use the gene symbols represented in the pathway to show how, in this case, what is the pharmacodynamics of this particular drug. There is a legend that you can click on that will show you all the different types of entities that might be in any particular pathway. You can always ask to reproduce pathway with permission from PharmGKB. There's a link right there that you can click on to request access. That's done very commonly. So the picture itself is a pretty picture to show you what's going on, but it doesn't really get into all the details. So of course there's a long text description underneath of that. These pathways, the image and the description, are often published in pharmacogenetics and genomics. If you scroll down to the bottom, you'll often see a citation for a particular pathway. You can see the components of the pathway by clicking on the side nav. In this case, you can see for every arrow on that pathway, the arrow from, what the arrow is to, and a PMID, or a literature source, at least one, for most every arrow on every pathway. Sometimes there are related pathways. In this case, there's a separate pathway drawing for the pharmacokinetics of warfarin. Go back. Any publications that were used in the creation of that pathway, you can find listing here. And you are able to download the diagram itself, the pathway in BioPax format, and some other formats as well. Okay. So the next thing I'm going to show you about how to browse are dosing guidelines. So these guidelines on PharmGKB are guidelines that provide prescribing information for particular drugs based on a patient's genotype. And there are a few agencies right now, or a few groups, who focus on creating these types of guidelines. And I'm going to tell you about two of them. On PharmGKB, both CPIC guidelines and a Dutch pharmacogenomic working group guidelines are listed. We also have some guidelines from a Canadian group and some professional societies as well. But briefly, I wanted to explain to you what CPIC and the other group does. So as I mentioned earlier, CPIC is the Clinical Pharmacogenetics Implementation Consortium. We have members from all around the world. And the goal of CPIC is to provide very detailed clinical practice guidelines for pharmacogenomics. And this is based on having the genotypes in hand. So it's assuming preemptive genotypes. All the guidelines published by the CPIC group are published in Clinical Pharmacology and Therapeutics, and they're updated approximately every 2 years. CPIC has a website at www.cpicpgx.org if you're interested in more information about that particular group. But I'm going to show you some of their guidelines as annotated on PharmGKB today. As I mentioned, in the Netherlands, there's also a group called the Dutch Pharmacogenetics Working Group. They are part of the Royal Dutch Pharmacists Association, and they also create prescribing guidelines or information about how to prescribe drugs based on a patient's genotype. And they have published a couple articles that are reviews, and you can check those out as well. So I'm going to talk to you about, I'm going to show you a couple examples of those guidelines as annotated on PharmGKB. Let's go back to the website. So we're still talking about browsing as well. So this is the landing page listing all the dosing guidelines that are annotated on PharmGKB. There's one column for CPIC, one column for the Dutch group, column for Canadian group, and so forth. You can organize these by clicking on the headers. You can filter it to just one particular source. If you're only interested in one particular source, then it orders the guidelines by that particular source, though of course you can still see the others. So we looked at the warfarin pathway, so I'm going to bring up, oh, I just messed that up. I'm going to have to redo that. We're going to have to edit that. Sorry. Can you make a note of that time? Sorry about that. Okay. All right. So you can order it by whatever group you want. If you pull down the menu, then the guidelines are ordered in that order. I'm going to look at a particular drug. I'm going to search for codeine. It narrows my list for me. You can see that there are 3 groups that have published some kind of guideline regarding codeine. And I'm going to show you what the CPIC annotated guideline looks like on PharmGKB. So as I said, these guidelines are for dosing based on a patient genotype, assuming that the clinician has a genotype in hand. So for example, we have a drop-down menu here where you could enter your patient's genotype. And you get a quick overview, summary, of this particular guideline's prescribing recommendations for that particular genotype. There's other information here as well about what the metabolizer status is for your patient, etc. And down here, the recommendation is, for this particular, for star 2, star 3 of CYP2D6, use label recommended age or weight specific dosing, so nothing in particular that's different. But as you scroll down, you can see there is a large table down here with lots of information of all different types of genotypes for this particular gene and what the dosing recommendation is for that particular. So if you want an overview of all the different genotypes, what it is that the CPIC group is recommending, you can scroll down to that table. We also provide, or you can enter a different genotype here anytime and get a different recommendation for that as well. We also provide for some of the guidelines right now. On PharmGKB, there's a short video about 2 minutes, 2-3 minutes, that sums up the content of the guideline manuscript and shows some other PharmGKB information that works with that particular guideline. You can also always access the guideline itself. You can download it, the actual manuscript and the supplement from this annotation page. Okay. And so the last group at the top that I'll show you how to browse are the drug labels, the annotated drug labels on PharmGKB. Taking a second to load. Okay, so just as with the guidelines, there are multiple agencies that issue drug labels. In the United States, of course, FDA, we have an FDA list here, but PharmGKB also curates drug labels from the European Medicines Agency, the agency in Japan in charge of drug labels, and also in Canada. You can see more information about the sources of drug label information from PharmGKB by clicking on this little question mark icon, and it'll tell you the different agencies that the labels come from. For example, with the Japanese Medicines Agency, we work with a group that has kindly translated labels for our site. Most agencies do not give any kind of recommendation for genetic testing on the drug label itself. Occasionally they do, but most of the time they do not. A question came up repeatedly for PharmGKB about what to do with a particular drug. What does, say, the FDA recommend? Do they recommend testing? Do they require testing? So there aren't exactly labels on those FDA drug labels right now to that effect. Testing required, testing recommended, actionable information. So PharmGKB came up with 4 different, zoom in on this, levels of pharmacogenetic or genetic information in test labels, and they've color-coded them. So it's divided into testing required, testing recommended, actionable PGX, or informative PGX. And you can read the descriptions here. If something has a red testing required label, that does not necessarily mean that the FDA is saying a genetic test is required, so just be aware of that. Could be some kind of protein or chromosomal testing. But we have used these labels to try to give people an overview, if I go back to the drug label landing page, of what each agency is recommending or what information each agency is putting on the label about pharmacogenomics, with respect to pharmacogenomics. And you can see across testing agencies, it may not be the same. Sometimes for the same drug, in the case of abacavir at the front row, 3 of these agencies have something about testing being required, and then another agency that barely even mentions anything to do with genetics. And it's like that across the board. Sometimes they're the same, sometimes they differ. You can filter these labels again by clicking, if you just want to focus on FDA or whatever. Some labels have dosing information in them. They get so specific they actually have some kind of recommendation of how to dose on the label itself. If you check that checkbox, you'll get a list of that right there. You can always type in your drug of interest and filter the list down to that as well. You are also able to download the entire table if you're interested in doing that from PharmGKB using this button. So I'm going to filter for codeine. We looked at codeine for the dosing guidelines. We're going to look at codeine here. If I want to see the label itself and the annotation on it, I'm going to click that. PharmGKB gives a brief summary of what's on the label, a little bit more detail about the label, usually some kind of excerpt. But you can also always download the drug label itself. I'm going to download that by clicking on this link right here and open it up for you. And you can see highlighting. So we took the actual FDA-approved drug label and highlighted all the places that had something to do with pharmacogenomics. So the highlighted drug label is a great thing to consult if you're interested in the content on that particular label. Okay. So there are other things that you can browse on PharmGKB as well. If you scroll down a little bit, you can see that we've kind of divided topics up into things that are more clinically focused and things that are more research focused. Dosing guidelines and drug labels, we also have links to browse at the top. They're listed down here. Pathways are also here. One of the things that are not listed at the top that I'm going to show you here are very important pharmacode gene summaries. They're called VIPs. And basically these are genes, a list of genes that are either very important in pharmacogenetics because they either metabolize a lot of drugs or because they're super important for the action of one or more particular drugs. There's not an easy way to browse through this. We're back to the alphabetical list here. But if you are looking for a particular gene of interest, again, you can use the sorting. CYP2D6 is a very important, I'm trying to find it here, there we go, gene for metabolism of drugs. Basically, these are in-depth summaries or full literature reviews on the gene, variants in the genes. It's a lot of text, not the most exciting thing to read on a website. There are individual summaries for specific, really important variants within that gene. And then also you can go to the actual published gene review itself. Again, these are published on most of the time in pharmacogenetics and genomics, just like the pathways are at PharmGKB. So the curators do the full lit review, they write the text, they publish them in that particular journal, but all the information is also available on the website itself. Okay. So the next piece of information, kind of data type that I'm going to show you on PharmGKB is called the variant annotation. I'm going to go back for 1 second. I want you to note this number. So there are almost 18,000 variant annotations on PharmGKB right now. So browsing 18,000 variants annotations are not really feasible. So if you come to this particular landing page, what you're going to get is an example list. There's a lot of information at the top if you want to read it about what these are, how they're created, but basically variant annotations are based on one piece of literature. The curator annotates the association between a specific variant in the gene and a specific drug or group of drugs, depending on the piece of literature. So variant annotation is the association between a specific variant and a drug and that drug phenotype. On this landing page, there's an example table at the bottom just to give you a feel for the kinds of information that. And we can show this, we can pop it out in full screen because this table is actually pretty big. In the table, we have a quick overview of 4 different variant annotations here. They list the variant itself by RSID, the PMID for the article that the information is annotated from, curated from, the drug associated with that particular variant, a brief summary about what the association is based on genotype or allele. So depending on what allele is represented for that particular variant, what the phenotype would be. The study size, a phenotype category, those include efficacy, toxicity, drug metabolism, or other. The gene that the variant is in, if it's not an intergenic gene, variant. And then some more information that's entered as free text by the scientific curators when they're annotating that particular association. So any one of these things, you can click the blue Read Now button and go to a page just devoted to that particular association between that variant and that drug from that article. But it's basically the same information summarized in a slightly different format. The interesting thing about having this full screen is that you are able to edit the columns however you want. So, for example, if you are interested in what the p-value is for the association, you can click the p-value and it now shows up here. If you want to know, are there cases versus controls, you can click those and you can see those columns being added. If you're not interested in that, you can click them away and they will go away. If you are signed into your account, your settings, the columns that you choose, will be remembered for you. And so if you really have a preference to how you like this table set up and what information you're interested in and what information you're really not, you can set that however you like and move off of this page and it remembers that. So I will show you, there are times that you make it to a list of annotations that are dozens long. And so being able to filter and sort by using these buttons on this tool is very nice to have feature. So the variant annotations are recorded by the scientific curator as is stated in the article. No judgment added, just the facts. Now I'm going to show you a different type of annotation called a clinical annotation. And at this point, the scientist gets to use some kind of judgment when creating these. So basically, a clinical annotation on PharmGKB is a summary annotation. So I showed you a table where there was associations between a particular variant and drug, but there may be many different articles that report an association between the same variant and the same drug. Sometimes, though, an initial finding is replicated. Sometimes an initial finding is refuted. Somebody else studies it and they don't find the same association. Sometimes some of the more well-known associations are reported dozens of times in the literature. Going through dozens of variant annotations like that to try to come up with your own kind of summary or overview of what the overall association is between that variant and that drug can be challenging. So the curators at PharmGKB essentially do that for you. They write these summary annotations about everything curated in PharmGKB, all the literature about a particular variant and a particular drug, and they sum it up and they give you a little bit of a judgment on it. So again, you can read more about these clinical annotations and how they were created. There's a whole publication on it, but I'm going to show you the summary table. Again, this table can pop out to full screen. It has the same buttons to be able to set the columns as you wish. But it's a little bit different column setup. So again, we have the variant in question. We have the gene that the variant is in, the chemical associated with it, or the molecule or the drug in this case, and the annotation type. But you don't get to see the meat of the annotation at this table level. You have to click down in order to see that. So I'm going to show you for an annotation about particular variant and simvastatin. So if you click the read now, this is where you get the genotype-based summary for all the literature curated in PharmGKB about this variant and drug. And it's genotype-based, as I said, so that means it's written each genotype, in this case, this is a CT variant, so there's a CC, a CT, and a TT, gives you a summary about what it means for that particular genotype and the phenotype related to the drug in question. Underneath, if you scroll down, you'll see all the variant annotations that were used to build this particular clinical annotation. So, in this case, we have like over half a dozen annotations that this clinical annotation, that this summary was based on. On the left-hand side, you will see a level of evidence. This level of evidence, if I click on it, it'll take you to a page that describes all the different levels of evidence in clinical annotation. And this is where the scientists at PharmGKB really get to use their knowledge and be a little bit subjective about judging the data that was published about the association. But in a strict way. So these levels do have strict criteria that are followed when applying a particular level to a clinical annotation. So the level 1A is the highest level of evidence, and that means basically that PharmGKB is aware of a place is actually implementing pharmacogenomics based on that particular variant and drug phenotype. Or there's a guideline published on it. So that's pretty much the highest level of evidence there is. If there are clinicians right now, they're actually acting on this information. The levels of evidence go all the way down to a level 4, which means there's either just like a case study or just some in vitro data, very, very tenuous information linking this variant and this drug together. And the level 3 level is assigned in places under circumstances in which some literature has reported that there's a positive that there is an association between the variant and the drug and a drug outcome, a phenotype. But there may be one or more pieces of literature that were unable to replicate that or refute that evidence. So in any cases where there's conflicting data, a level 3 will be assigned. And again, you can read through this to familiarize yourself with the criteria at every particular level, but they are every single clinical annotation. Back one more time. Every single clinical annotation in PharmGKB will have a level assigned to it. In this particular example table on the browsing page, only level 1A, and maybe level 1B, yes, level 1 evidence is displayed. Okay. There are a few other things on this landing page that I will just briefly show you on the home page. There are these resource buttons. These are pages on PharmGKB that focus on a particular area. The Cancer PGX page is just a landing page on PharmGKB for everything that has to do with cancer pharmacogenomics that's contained in the PharmGKB resource. PharmGKB does not focus on cancer pharmacogenomics per se, especially somatic cell mutations. There are other resources that focus on that. But PharmGKB does have some information on the pharmacogenomics in those cases, specifically drug labels with that kind of information on it. And then, of course, we also have germline annotations for germline variation that influences cancer drugs. So all of that can be found linked to from that cancer pharmacogenomics page. And even though PharmGKB itself is not an implementer of pharmacogenomics, we do collaborate with several groups. And there's a list on this particular page for implementation resources for pharmacogenomics. And so that is a really good place to go if you're someone in the clinical setting that is looking for resources to help you actually get the information available on PharmGKB implemented in your setting. The gene-specific information tables are really great resources to go along. They go hand in hand with the CPIC guidelines that were talked about earlier. And this is just a quick stop to access tables that are really key to being able to implement the CPIC guidelines for those particular genes. Okay, so now I'm going to talk to you about searching on the PharmGKB website. So if there's something that you know that you want to find, a particular piece of information, here's how you would go about doing it on the website. The main search box on PharmGKB is a Google type search. You just type in whatever information you're looking for, and it will do its best to bring back pages on PharmGKB that are relevant to what you want. It really works best for genes, drugs, and diseases, those entities in PharmGKB that are kind of the core entities. I'm going to show you an example if you search on codeine. So if I search for codeine, there is an automated drop list that tries to figure out what exactly I'm searching for. I can either select from this list, or I can just enter whatever I want and hit the magnifying glass. If I hit the magnifying glass, I come to a search results page. Again, it kind of lists what was in that drop-down box, but now it tells me what kind of information, dosing guidelines, drug labels, annotations are available for each one of these entities, these options that it's giving me for codeine. There are a lot of them. You can see showing 1 to 25 of 147. If I didn't know specifically what I was looking for, I could narrow it down by saying, okay, I'm interested in some kind of pathway that has to do with codeine. I would click that filter, and then I can see there's only one in that long list of 140 returns that was actually a pathway. Drug labels and dosing guidelines. Again, so you can filter the list however you want. But if I wanted to know everything in PharmGKB that has to do with codeine, I would go to the molecule, the drug page for codeine, which is going to be the first return. Click that. This page setup with the side nav and the numbers at the top are on every single drug page and gene page, phenotype page in PharmGKB. They're all set up the same way. If I was on a gene page, I'd have a little bit different icon up here, and it would have a gene symbol instead of a drug, but the page would be relatively set up in the same way. The first tab that you come to when you come to a drug page like this is the Overview tab. If available, it gives me some kind of structure and a little bit about the alternate names for this particular drug. The blue circles indicate that there is information behind that particular tab. So, for example, if I go to the Drug Labels tab, up here I could click this as well. These are going to give me the same returns, but I happen to know there's 3 here if I look here. If I go to the Drug Labels tab, it gives me all the drug labels that are annotated in PharmGKB about this particular drug. So we have 1 label from the FDA, 1 label from the Canadian group, and 1 label from the Japanese Medicines Agency. I can read any of those. I click on it. It gives me the annotation in PharmGKB. Again, I can download the highlighted drug label by clicking that link. But now I'm only seeing the drug labels as opposed to when I was browsing and I saw all the drug labels available from all the agencies. I'm on the codeine, molecule, or drug page, so I only see the labels that have to do with codeine. Same with variant annotations. So for example, this is what I'm talking about. I was mentioning earlier in the browsing section of this talk that, that you could have a lot of different annotations in one of these tables. I'm going to show this table in full screen and you can see what I mean. So trying to look through this entire table could be very tricky. We know that everything has to do with codeine in this table, but maybe I'm only interested in maybe I'm only interested in genes such as, in this case, UGT2B7. So I'm going to go to the filter box, UGT2B7, and then the table will shrink for me to show me only the annotations that have to do with that particular gene. So again, being able to pop these out into full screen and being able to edit the columns comes in handy in cases like this where you can have many, many annotations for a particular gene or drug. That was the variant annotations table. Clinical annotations, same way. We have fewer clinical annotations than we do variant annotations. That's always the case, but still sometimes nice to show it in full screen. All publications that were annotated in any way regarding codeine can be found on the Publications tab. Pathways containing codeine on the Pathways tab. The Related To tab is a tab which gives you a summary of all information that we have about codeine and any other entity, any other drug, any other molecule, or any other phenotype. And you could scroll through that list or filter, just like anywhere else on the website. We do have a Links and Downloads tab on every drug and gene page. If you're interested in different nomenclature or interconnecting with other databases, other NIH resources, the Links and Downloads tab have links to other resources and the IDs for this particular gene or drug in that particular resource. These links will take you off of the PharmGKB website. If I go to the Prescribing Info tab, what I get on that particular tab are all the drug dosing guidelines from the different groups, CPIC, the Canadian group, the Dutch group, or any professional society. Any dosing guideline that has been annotated in PharmGKB, you can pull up, including the video, including a drop-down menu that lets you select the patient genotype. But also on the Prescribing Info tab, we have something called RX annotations. This is one more type of data annotation that I didn't mention before. There's more information about this if you want to read about it, but basically what it is that oftentimes there are studies done where the author may come up with some kind of recommendation for how to use a particular drug, for how to prescribe a particular drug, but it's not a guideline that's authorized by a particular agency or society or group. And so, so those you will find underneath on any gene or drug tab, on any gene or drug page on the Prescribing Info tab if it's available on PharmGKB. Okay, so that's just an overview of a drug page, but I've still got codeine up here in my search box. I'm on the codeine page. Maybe I'm interested in finding, all right, there's a lot of information on codeine. I went there, I saw a ton of information. So I'm interested, well, does PharmGKB have anything about codeine and warfarin together? Like, is there any association between codeine and warfarin? Well, I start typing warfarin in this box, and you can see it already says no results match your query. So there is no information. So I can try to do, it's basically a joint, it's an and. Search on PharmGKB for these two drugs together. There is nothing available for that. So I know that right away. But maybe I want to try something else. I'm typing in a gene now, ABCB1. I can see that there is an option for ABCB1. This page is a page that gives me just the information and annotations that regard both of these together. So now I'm on a combination page, codeine, the drug, and ABCB1, the gene. So the information presented on this page, you can see there's no dosing guidelines that have anything to do with both codeine and ABCB1 genotypes or drug labels, but there are a couple clinical annotations, and in the pathway, the codeine pathway, ABCB1 is mentioned. That is why it's being returned here. There are some variant annotations where you have ABCB1 and codeine, an association between them in an article. If I remove ABCB1 and I put a different gene up here, step 2D6, I want to look at the combination between that gene and that drug. Now I see I do have drug labels and dosing guidelines available for that combination as well. If I wanted to start all over again, say, okay, I understand there's information associating codeine and these genes together. There was nothing linking codeine to warfarin, but I'm just interested in warfarin now. Just be aware that when you use the search box at the top, you do need to clear out all the current searches if you want to start a search over again from scratch, like say for warfarin. Now I'm on the warfarin page. Okay, so basically the search in PharmGKB allows you to do combinations of genes, drugs, and variants as well. I could type an RSID in there, and you will get a page that has just the intersection of the information between those entities. In this segment, I'm going to talk about how to download data and download information from the PharmGKB website. The quickest way to download large chunks of information from the PharmGKB website is to use this Downloads button at the top of the homepage. All the data sets available on PharmGKB are linked to from this particular page. The top section that talks about primary data allows the user to download the major entities on PharmGKB, such as genes, drugs, variants, phenotypes, as they're annotated in PharmGKB. So if you're interested in all the genes that are annotated on PharmGKB, this genes download is where to go. There's like a one-sentence summary for all of these files about what's contained in it. There's also a little blue eye icon next to these. You can click that icon and get more information about what it is that you would be downloading before you download it. So it gives you kind of the structure of the file, what information is contained in the different columns of the file. So it's a pretty good resource to look at because sometimes there's misunderstandings about what is exactly contained in these downloads. So for example, the genes download on PharmGKB is not meant to be a download of every single gene identified in humans. Likewise, the download of variants is not every single variant that's contained in DBSNP. It is variants using a DBSNP ID that have been annotated in PharmGKB and so forth. So if you look at the little blue icons, it's really helpful so that you can make sure that you're getting the information that you want and making sure that you understand the information that you're getting. Okay. All of these downloads will tell you which drug labels are annotated or which genes are annotated or which variants, but you don't get the variant and clinical annotations that were discussed earlier today themselves when you download these primary data files. If you go to the annotations data section of the downloads page, that's where you would get that information. So these files are more complex because there's so many. If you'll recall the tables for the variant annotations and the clinical annotations that were shown on the website, there's many, many columns. There's many different relationships between the information in the different columns. So the downloads of that type of information are not simple, just kind of one file type things. They're a zip drive of multiple files that you have to kind of navigate. So we have example annotations data that you can download so that you can see what type of data this is. There are two main ways to get to this annotation, to get to these annotations. The variant and clinical annotations data is all the information on the website, every single piece of information that was written by the curator about a variant or a clinical annotation. I'm going to click this request. In order to get this data download, you have to ask for it from PharmGKB. It doesn't just download automatically. So I'm going to click here. And it's going to take you to a licensing page. All data is free at PharmGKB for academic, not-for-profit uses. Requesting a license in that case is very simple. You just need to fill out the form. You say you're requesting annotations or relationships. I will go into that in a second. For academic purposes, an email gets sent off, and then someone contacts you with how you can access that information. Non-academic licensing is a little bit different. That does have to go through Stanford's OTL office, and so that's a little bit of a different system. But for academic purposes, it's pretty simple and straightforward to get a license. You can see all of the people that have a license already. If you'd like to, you can hit this list of annotation licenses. It gives you their name, their institution, and something about how they were going to use the data, because we do ask you for a summary or an abstract about how the data will be used. If you want more information about the particular files in that download, we have all that information available. This gives you a description, a long description of every column of every file in the download, in addition to giving you the example on the previous page. So you can get basically a README file of everything that's in the download and then an example data in there so that you know exactly what you're getting. The difference between the annotations and the relationships data is that the relationships data also includes all connections between genes, drugs, and variants in PharmGKB from other sources, such as drug labels, the dosing guidelines, annotated literature, in addition to those variant annotations and the clinical annotations. So the annotations file gives you all of the variant annotations and clinical annotations on PharmGKB. The relationships files give you the same annotations plus other connections or links if you think about it that way, between the drugs, variants, and diseases. PharmGKB also has, for download, consortium files that have been published. So if you see in a particular publication where PharmGKB is referencing a download, then you would come to this downloads tab and you'd be able to link it up by the article, the PMID. You can find the data set of interest that way. And also some other data sets that are made available through PharmGKB. The most sought after information is the annotations data, and I just want to reemphasize the fact that it is free, and there is one step for you to go through to request the license, but it's a pretty painless step. We are not trying to make it painful at all to get the information from the website. Okay, so that's basically an overview of the entire website, PharmGKB. If you have any questions about PharmGKB, the website, or the data contained in the website. Please do use that contact button at any time, and please do include an email. It's not required because we do want to be able to encourage people to send feedback, comments about the site anonymously if they prefer. But if you have questions that you'd like an answer to, please be sure to put your name and your email in the feedback form as well. Thank you.